Okay.
Yeah, can you please tell me about yourself?
Okay, myself Kritika Patil. I am from Burhanpur and I have completed my graduation B Pharmacy from Thakur Shiv Singh Memorial Pharmacy College with 8.39 CGPA. I am quick learner and hardworking. Now I am excited to start my career in clinical research where I can contribute my skills and knowledge to enhance healthcare system and enhance patient well-being.
Okay. Okay. Like, what is your strength and weakness? Can you please tell me?
Um, Time management and teamwork are my strengths,
Okay.
and weakness is overchecking work.
Okay. So, if you have team of 5 members, okay. So, how do you manage your team?
Communicate with each other, listening others' idea too, and then uh, tra- maintaining the task individually.
Okay. Hi, Kritika.
Hello, ma'am.
Okay. Would you like to tell me what is an IND?
IND is investigational new drug. And after preclinical trials, we feel application INDA, investigational new drug application, which is a formal request to a regulatory authority for permission to conduct clinical trials.
Okay. What is uh what is NDA?
NDA is a new drug application which is filed after phase 3 and submitted to regulatory authority to approve the drug to market it.
Okay. Can you explain me what is phase zero?
Phase 0 is basically microdosing phase, in which in this phase we gather preliminary pharmacokinetic data. It is not a mandatory phase.
And what is the participant number and the time span for phase zero?
It is time span is likely less than 7 days, and participants are 2 to 10.
Okay. What do you mean by maximum tolerated dose and how it is calculated?
A maximum tolerated dose is the highest dose which doesn't cause any unacceptable side effects, and we can calculate it by single ascending phase and multiple ascending phase. In single ascending phase, we give a small dose to a group of patients, then we slightly we increase the dose and give it to another group, and this process steply in this process continue in stepwise manner. Then the doctors reach to the highest maximum tolerated dose, then we stop this process and multiple ascending phase is started.
Okay. Can you tell me what is the major difference between SAD and MAD?
Sad and mad, single ascending phase. In this phase we increase the dose, and we increase the dose till we reach to maximum tolerated dose. After when we reach to maximum tolerated dose, then MAD starts, multiple ascending phase, when we gives the some multiple doses to a patient. In single ascending phase we give single dose to patients.
Okay. What is Cmax and Tmax?
Cmax is a peak concentration of drug in the blood, and Tmax is a maximum time, nahi, time at which the concentration is maximum.
Okay. What is half-life elimination?
Uh, it is a time when the half of drug is eliminated from the body.
Kratika, can you
Okay.
please go
Can
through all the phases of clinical research?
Yeah. Phase 0, microdosing phase. In this phase, 2 to 10 participants, healthy volunteers are involved. In this phase, we gather only preliminary pharmacokinetic data. And then phase 1 is basically defines maximum tolerated dose, route of administration, in safety, in which there are two phases: single ascending phase and multiple ascending phase. And phase 1 is also called human pharmacology phase. And then phase 2 is a exploratory phase, therapeutic exploratory phase. In this, we check dosing requirement, safety and doses, in which there are also two phases, phase 2A and phase 2B. In phase 2A, we calculate the how much drug is given, and phase 2B we calculate how well the prescribed drug is work. And phase 3 is therapeutic confirmatory phase. In is there are also two phases, phase 3A and phase 3B. Phase 3A, in which we compare the investigational new drug with standard treatment, means with gold standard treatment. And phase 3B is for conducted for commercialization and gather more data.
Okay.
And phase 4 is done after NDA submission. In it is a post marketing surveillance phase, in which we detect the rare side effects.
Okay. What is CPU?
CPU, CPU is basically central pharmacological units where pharmacodynamic studies are conducted.
Okay.
Tightly, tightly, tightly controlled units.
Okay. What is the difference between pharmacokinetics and pharmacodynamics?
Pharmacokinetics is the what the body does to the drug, means absorption, distribution, metabolism, and elimination. And dynamics means what the drug do with the body, means pharmacological effects and any side effects.
Okay. Can you please tell me the standard definition of clinical research?
Okay, any investigation in human subjects intended to discover or verify intended to discover or verify clinical, pharmacological, or any pharmacodynamic effects of an investigational product, or to identify any adverse reactions to an investigational product, and to study the absorption, distribution, metabolism, and elimination of an investigational product with the object of its ascertaining safety and efficacy. Clinical trial and clinical study are synonymous.
Okay. So can you please let me know what is absorption?
Absorption is basically how drug reaches to site of administration to bloodstream.
And what is metabolism?
Metabolism is basically how drug breakdown in the blood.
Okay. What are the difference between like phase 2 and phase 3?
In phase 2, we determine the safety and efficacy, and phase 3 we compare the drug with standard treatment. Means in phase 3 we check effectiveness, and phase 2 we check for safety and doses.
Okay. Okay, Kratika, your overall knowledge is good and you like the overall interview was good. Thank you for joining.